So right now, this is a big board to welcome Dr. Kumar. Okay, please.
Thank you, Professor. Good evening, professors and my colleagues. Today I would like to give you a brief account about the recent research and development of Antrodia cinnamomea, endemic medicinal mushroom which is native to Taiwan. As we know that the mushrooms are rich nutritive food and it contains a lot of fibers, minerals and vitamins. Data from the British Mycological Society, they reported that there are 1.5 million fungal species are identified and 5,000 are mushroom and truffles. Among them, only 2,200 mushrooms are edible and consumed by humans worldwide. However, very few number of mushrooms are identified and characterized as the medicinal mushrooms. Among them, these 5 mushrooms received much attention and very popular as a medicinal mushroom, especially the Ophiogordypsis sinensis. This mushroom, also known as the caterpillar fungus, which is native to the Tibetan plateau or the northern part of China. And this mushroom is used as in traditional Chinese medicine over a millennia. And this is the most expensive medicinal mushroom in the worldwide, which cost $40,000 to $80,000 per kilogram. And next, the costliest mushroom is Antrodia cinnamomea, which is cost about really $15,000 to $25,000 per kilogram. This is native to Taiwan, and it is the rare and endemic species. Both these, the Ophiogordypsis sinensis and Antrodia is well characterized medicinal fungus in terms of their high content of secondary metabolites or bioactive compounds, especially triterpenoids and polysaccharides. Since the Antrodia cinnamomea is used as a traditional Chinese medicine over a millennia, but it come to the real world after 1770s. It holds an interesting story. In the year of 1773, a famous Chinese physician, Dr. Wu Sha, he is relocated from Fujian province of mainland China to the Taiwan, and he settled in the eastern part of Taiwan. It's currently the Hualien and Yilan city. At the time, the eastern part of Taiwan is occupied by, inhabited by ethnic people called Kawalan people. These Kawalan people have a unique cultural ritual habit that during the cultural festival they drink a lot of alcohol and they easy to hangover. One day the people invite Dr. Wu Sha to the cultural festival, and that time the people are drunk and hangover, and the village healer or the wise man gave some decoction to the people. The people easily recover from the alcoholic hangover. So the Dr. Wu Sha is really interesting about the medicine, and the village man said this is a mushroom collected from the wild, and Wu Sha extensively surveyed whether the Antrodia cinnamomea or the mushroom nutrition have only the alcoholic hangover effects or it have any other effects. Luckily, he found a lot, and the village man said that the Antrodia is not only for an alcoholic liver disease; it also have several other health effects, including it can be used for intoxication, diarrhea, abdominal pain, fever, headache, and even though for cancer. After using several years, this mushroom is considered one of the best hepatoprotective drug in Taiwan, and the recent research in past 2 decades suggests that the medicinal application of this mushroom is go far beyond the traditional usage, as evidenced by the number of publications in the peer-reviewed international journals. The first papers appeared in 1995. The authors describe about the chemical constitutions in the mushroom, and after 2000 the publication was significantly increased and reached in the peak in last 3 years, especially in 2011, 2002 and 2013. There are average 35 publications per year. Since Taiwan is the major contributor of the Antrodia cinnamomea research, they published more than 200 articles, and some other countries like Australia, Canada, Japan, Netherlands, Korea and the USA also contributed in Antrodia cinnamomea research, and they published a few papers. Most of the research are focused the pharmacological effects or the health effects of the Antrodia cinnamomea, and considerable publications also demonstrating the chemical constituents and production and improvement of the Antrodia cinnamomea, and the taxonomical or phylogenetical analysis, pharmacological effects. Since the Antrodia cinnamomea is recognized as one of the hepatoprotective or liver protective agent, so the researcher want to know how the Antrodia protect liver from the toxic or toxic chemical induced liver injuries. As we know that the liver is the largest autonomous body organ which detoxify toxic chemicals from the circulation. There are several factors affect the liver injury, especially excessive alcohol consumption, drug and chemical intoxication, and even the viral case affect the liver. The recent studies showed that the excessive alcohol consumption induced fatty liver formation, liver fibrosis, and oxidative stress in the liver. By fatty liver synthesis and triglyceride accumulation can induce fatty liver, and excessive accumulation of extracellular matrix proteins can induce liver fibrosis and cirrhosis, and free radical and reactive oxygen species can cause oxidative stress in the liver. The recent study showed that both mycelia of Antrodia cinnamomea and fruiting bodies of Antrodia cinnamomea significantly protect the liver from fatty liver fibrosis and oxidative stress through the induction of antioxidant genes or to the downregulation of matrix metalloproteins and the transforming growth factor activities. The Antrodia cinnamomea not only protect the liver from the alcohol-induced liver injury, it also protect from chemical intoxicated liver injury. In this study, the various researchers used the carbon tetrachloride as a chemical to induce liver injury. The carbon tetrachloride induces lipid peroxidation and liver fibrosis through the accumulation of pro-fibrogenic mediators and pro-inflammatory mediators. The research shows that fruiting bodies of Antrodia cinnamomea significantly inhibit the carbon tetrachloride-induced liver injury through the downregulation of matrix proteins and transforming growth factor and the collagenase, or to the downregulation of lipid peroxidation. Further studies shows that the not proved extract of Antrodia cinnamomea have these liver protective effects. Their compounds also have these effects. In particularly, the anthraquinonol and the anticin C isolated from the mycelia of Antrodia cinnamomea inhibits alcohol and free radical induced oxidative stress through the upregulation of antioxidant genes. And another study showed that the ibricoic acid and the ibricoic acid significantly protected rat liver from the carbon tetrachloride-induced liver injury through the induction of antioxidant and the inhibition of pro-inflammatory genes. Another study from the Vietnam, they used the cell-free analysis and found that Antrorin A and Antrorin C inhibit hepatitis C virus proteases. So the hypothesis that this drug can be used to inhibit hepatitis C virus replication. And another study shows that Antrodan, a long-chain polysaccharide from Antrodia cinnamomea, can inhibit LPS-induced inflammation, liver inflammation in rat through the downregulation of pro-inflammatory genes. The liver protective effects of Antrodia cinnamomea is not limited to the liver protection. It also extended to anti-cancer effect against the hepatocarcinoma cells. Both pure compounds isolated from the fruiting bodies and the crude extracts, especially the anticin A and anticin C and the methylancinate A, significantly inhibit the growth and proliferation of hepatocarcinoma cell lines, and the anticin B and methylancinate A and methylancinate B induce apoptosis of hepatocarcinoma cells through the upregulation of reactive oxygen species and the induction of caspase signaling cascades. And beside, another study shows that the 4-acetyl androquinone B from mycelia of Antrodia cinnamomea induce apoptosis, as well as it inhibit the cell cycle progression through the modulation of cyclins and cyclin-dependent kinases. The anti-cancer effect of Antrodia cinnamomea is not limited to the hepatocarcinoma cells, and it also extended to other cancer cells, including prostate, leukemia, bladder, colon, oral, lung, and the pancreatic cancer cells. The anti-cancer effect of Antrodia cinnamomea is especially through the induction of cell cycle arrest and inhibit proliferation and induce apoptosis. Some further analysis showed that Antrodia cinnamomea inhibit the endothelial cell proliferation and neovascularization of endothelial cells, because the neovascularization support tumor cell development. And another studies also show that the Antrodia's pure compound or crude extract of Antrodia cinnamomea inhibit migration of tumor cells, and even though it can modulate the epithelial to mesenchymal transition. The accumulated studies of Antrodia cinnamomea shows that the Antrodia cinnamomea is various health, biological or pharmacological effects. It can be used for anti-inflammation. The professors detailedly described it before. It also have anti-inflammation, anti-cancer, anti-diabetic, and hepatoprotective, neuroprotective, and cardioprotective effects. As a result, there are more than 35 Antrodia cinnamomea based health food supplements are available in the market in the form of tonics, capsules, and powder. And the Antrodia cinnamomea based health food industries also proposed some new idea that it can be easily available in the daily consumable foods, like candies, drinking health drinks, and it can mix with green tea, and also we can use with like a dip tea. Excuse me. However, the research of Antrodia cinnamomea faced a lot of challenges. There are numerous bottlenecks, especially the classification and the nomenclature of Antrodia, and the major source, especially the abundance, because the Antrodia cinnamomea is have the host specificity and very rare in the nature, and also a slow growth. And the large production also, the researcher also faced some major challenges in the large scale production. The nomenclature is similar to the discovery of Antrodia cinnamomea. The nomenclature and the classification of Antrodia cinnamomea also have a long chronological history. Before 1990s, the Antrodia cinnamomea is known as Pyrophormus formosana. In 1990s, Zhang and Su, they named the Antrodia cinnamomea. So they named the new change as Ganoderma camphoratum, because the morphological and the physiological characters of this mushroom is closely resembled to the Ganoderma species. Therefore, they named as Ganoderma camphoratum. But 5 years later, in the year of 1995, Chang and Cho, they proposed a new name, that Antrodia cinnamomea, because the characteristics are closely resembled to the genus Antrodia. and the cinnamomea is the host plant, so they give a new name Antrodia cinnamomea. Then 2 years later, Wu and his co-worker, they give us an idea that the Ganoderma camphoratum and the Antrodia cinnamomea have some conspecific. So a new combination or a new name was given, that is Antrodia camphoratum, and they said this is the most appropriate name for this fungus. Until 2004, the mushroom was identified as the Antrodia camphoratum, and 2004 again the Chang and Cho, they suggested that Antrodia cinnamomea is most preferable and most concise name for this fungus, because the holotype was submitted to Taiwan Forest Research Institute. In 2004, the phylogenetic analysis based on ribosomal subunit sequence and ribosomal RNA genes indicate that this fungus is distantly related to Antrodia genus. So the fungus is moved to a new they formed a new genus and moved to a new genus named Taiwan fungus, and provided a new name Taiwan fungus camphoratus. Again, in 2007, the Chu studied the polymorphism analysis with the transcribed spatial region of the ribosomal RNA. They conclude that the mushroom is not belongs to the genus Taiwan fungus. It is belongs to the Antrodia genus Antrodia. Finally, they classified this mushroom as classified, and it is belongs to the family of Formitopdaceae and the genus Antrodia, and the species names or Antrodia cinnamomea. Next, the host specificity. As I told you before, that the Antrodia cinnamomea is a unique mushroom which grows inside the age-old tree of cinnamomea, Cinnamomum caniharia. This is also called a bull camphor tree, which is also endemic to Taiwan. It's a large evergreen tree, can be found in the Taoyuan, Hsinchu, Taichung, Nantao, Hualien, and the Kaohsiung mountain regions. Due to this tree is not only important for the source of Antrodia cinnamomea, it is also very important because of the high quality wood furnitures. These woods have a unique scent because of high content of terpenoids. Therefore, to conserve the tree, the Taiwan government passed a bill that it is prohibited of cut down of this tree. So the researchers are urged to found a new way to produce this mushroom. They found three major techniques like cut-log cultivation, solid-state cultivation, and liquid fermentation. In these three are artificial cultivation system, and this is the natural cultivation system. In these four different culture conditions, the growth time, terpenoids, secondary metabolites, and the toxicity are totally varying. For example, the growth time for the natural growth condition is 2 to 5 years. For the cut-log, it takes 2 years, and solid state 4 months, and this is only 2 to 4 weeks. But the terpenoid content was varying, and there are very less terpenoids was found in the liquid fermentation. Because this mushroom is rich in unique chemical composition. More than 80 compounds were identified from this mushroom. The majors are terpenoids and benzoic compounds. Therefore, the researcher developed a new strategy to increase the production of the secondary metabolites. In the year of 2011, Lu and their team proposed a new strategy using the artificial intelligence-based techniques to optimize the fermentation condition to produce triterpenoid. In this study, they used two optimization models. One is the response surface methodology and artificial neural network. The content of triterpenoid was analyzed with the genetic algorithmic As a result, they found that the maximum terpenoid production was achieved with the medium component condition of glucose 25.2, peptone 4.48, and soybean flour 2.75 gram per liter. The following year, the next other team by Young and their team found that the substrate of some specific substance to the culture medium can produce a higher number of terpenoids and biomass and even the polysaccharides. In the study, they used citrus peel extracts. As a result, compared to control, the supplementation of citrus peel extracts, especially the pomelo, lemon, orange, and grapefruit, significantly increased the polysaccharides and the terpenoid content compared to control. Especially the lemon peel supplementation of lemon peel extra showed the better increase of triterpenoids and polysaccharides. Next, the other team by Zhang, they found that the optimization of pH of the culture condition may chance to increase the production of secondary metabolites, especially the androgen C compound. They found that in particularly the initial pH of 5 and the final pH of 3.67 showed a better increase of androgen C in some. Better androgen C production. Next, the other team by Chan et al. They found that to enhance the 4-acetyl androquinonol B, which is one of the important compound in Androdia cinnamomea, to increase this production, they used the precursor of this compound, especially the volatile compounds. The HPLC analysis showed that there are number of volatile compounds were identified in the Androdia cinnamomea culture filtrate, especially the especially the 2,4,5 trimethoxybenzoaldehyde and nerolidol. The supplementation of nerolidol and TMBA significantly increased the production of 4-acetyl androquinonol B in submerged culture. Next, an interesting study done by Wang and their co-workers. They found that culture of Androdia cinnamomea with deep ocean water significantly increased the production of triterpenoids, polysaccharides, and total flavonoids compared to the reverse osmosis water cultured Androdia cinnamomea. This study also shows that Androdia cinnamomea cultured with deep ocean water not only increased the production of triterpenoids and polysaccharides, it also enhanced the hepatoprotective effects of this mushroom. As the histopathological analysis with rodent model shows that this is normal, and the animal was treated with thioacetine to induce liver fibrosis, and they treated with various drugs like silymarin, deep ocean water alone, and deep ocean water treated with cultured in the Androdia cinnamomea in 1x the 2 doses. They found that compared to the drug-induced control, the liver fibrosis was significantly inhibited by Androdia cinnamomea, which is cultured in deep ocean water. And compared to the ROS water, the effect was largely observed in the deep ocean water cultured Androdia cinnamomea. Next, the collagen accumulation is one of the biomarker of liver fibrosis, and gave a positive feedback that compared to the reverse osmosis water, the deep ocean water significantly increased the production, increased the protective effect. Okay. Next researchers also interested to increase the secondary metabolites in solid state condition. In this study, Wang and their co-workers they used 4 different strains of Antrodia cinnamomea, especially AC3, AC5, AC7, and AC9. The second digit is encoded age, that means 9-month-old fruiting body, and the single strain with different age also investigated, like AC9 was cultured in 3 months, 6 months, 12 months, and substrate with other species like cinnamon camphoratum. The data, the HPLC profile shows that the chemical composition was totally different in each strains, because, for example, the andesine K was largely observed in the strain of AC9, it is very low in AC5. So the chemical profiles are largely depend on the strains, and largely depends on the culture age, because the second digit culture age 3, 6, 9, and 12, the andesine K was largely observed in the 12 months age old culture. Okay. Next, to take the Antrodia research in the next level, researchers identify and characterize some genes, metabolic involved genes, which can largely involved in the synthesis of secondary metabolites. For example, the lipase gene was largely observed in wild fruiting body compared to the artificial culture. The first line indicates liquid mycelia, and the solid state mycelia and solid state fruiting body. The highest was observed in natural basidiomyces, and the recombinant lipase gene and substrate with trilinolene significantly produce a new compound called linolenic acid in the culture. Then the other studies also found a novel lipopolysaccharide gene, which also help involved in the synthesis of secondary metabolites. And another study, they found that cloning and characterization of the linosterol protein alpha dimethylase, which is involved in the synthesis of steroids. They found this gene was abundant in the wild fruiting bodies, and somewhat found in the liquid culture submerged mycelia. Then they characterized compared to the submerged mycelia, the aerial mycelia was significantly expressed this gene. Next, a recent paper published in PNAS in the year of 2014, they obtained a 32.15 megabyte genome draft containing 9,254 genes. Most of the genes are involved. Most of the genes are involved in the biosynthesis of MAV-alertan pathway, triterpenoid pathway, cytochrome P50 family pathway, and polyketone synthesis. They compare, these are the major pathways involved in the biosynthesis of secondary metabolites, and the histogram shows the number of genes involved in particular biosynthesis, which is correlated with the KEGG subgroups. In this study, they used two strains, mononucleate Antrodia cinnamomea strains and solid state mycelia and wild type fruiting bodies. The high number of metabolic genes was found in the natural basidiomycota, and these stars indicate the number of genes identified by this study is well correlated with the pathway involved in MAV-alertan and DOX-P pathways. Next, the toxicological assessment of different scientists also proposed that the mycelial extract isolated from the submerged culture and the fruiting bodies extract was given to the rat and mice for subchronic 90 days, and they found there was no any lethal effects and body weight loss, no any hematological effects, and there was no changes in the serum biochemical parameters. Also, there was no abnormalities in the internal organ. This data strongly suggests that the consumption of androdia is highly safe and easy, and it's no effect to the human body. The other preclinical trials with chicken also show that the specific pathogen-free chickens fed with androdia extract for 35 days found that there was no changes in the biochemical or physiological parameter. So they found that the ACA could be enhance the immune function of the organ of pathogen-free chickens, and it could be an attractive application for nutraceutical and pharmaceutical interest. Thank you. Thank you for your kind attention.
Well, thank you very much. Thank you so much for Dr. Kumar extended talk related to the arthritis, pneumonia, many issues, such as like culture and biochemistry and pharmacology and so on. So I think it's very detailed in Howard's detailed studies on this mushroom. So maybe any question? I open one to